Good afternoon, everyone, and welcome to the second edition of the EUGLOH 3 Minutes PhD competition, organized by the University of Ludwig Maximilian, University of Munich, University of Porto, and University of Alcalá. The objective of this event is to challenge doctoral students to explain the research work within a limited time of 3 minutes. As you know, it consists in two phases, the internal one that was held in a university to choose the two representatives, and the final phase, which is being held today at the University of Alcalá, with the participation of 17 students from the universities of the EUGLOH Alliance. Regarding the contest rules, participants have 3 minutes to present the research topic, the objectives, problems identified, contribution to the solution, and the impact of their work. Please remember not to mention your institution name. As you know, it's even not allowed to place the logo on the slide. Only a static slide projection may be used as supporting element. Interaction with the audience that involves active dialogue is between the two parties is not allowed. However, posing rhetorical question is not considered interaction. Participants who exceed this time will be disqualified. You will have the timer, as you have seen this morning, in front of you, and it will turn into red when 30 seconds will be left. The jury consists of 5 members: Joaquín Luis Faria, full professor from the Department of Chemical and Biological Engineering at University of Porto; Marotti Laszlo, project manager for research, development, and innovation at the University of Szeged; Julia Seron, research director of the Munich Science Communication Lab at Ludwig Maximilian University of Munich; Mercedes Díez-Prados, associate professor at the Department of Modern Philology at the University of Alcalá; and Francisco Manuel Saez de Adana Herrero, full professor at the Department of Computer Science at the University of Alcalá. The jury will make its verdict based on different criteria. On one hand, the understanding and content, clarity, coherence, relevance of the objective and results, language, illustrative information and data, and time distribution. And on the other hand, engagement and communication, interest created by the topic, enthusiasm, ability to maintain the audience attention, security and confidence in the speech, clarity, and readability. In total, 70 students are participating today. The intervention will be carried out following the order established by the draw, as you have seen the list placed in the entry. We will be naming the participants. And the first one, you are now prepared. So thank you for your attention and good luck to everyone. Thank you.
Picture yourself at the doctor's office. You're told the treatment worked and the cancer is finally gone. You can now make plans and enjoy life again. However, years go by and all of a sudden it's back. And this time it's worse. It has spread to other organs in your body. My PhD is dedicated to this moment, how it happens, and in a new strategy for us to prevent it. This spread of cancer is known as metastasis, and it is the leading cause of mortality amongst cancer patients. The problem is quite a long time can separate the arrival of a cancer cell in a new organ and life-threatening disease. For example, in breast cancer, metastases can appear anywhere from 2 years to more than a decade after successful treatment. And this happens because once in a new organ, cancers don't grow right away. They pause and stall, turning this into a Cold War. As spies sent deep into enemy territory, cancer cells hide and lay quiet. Our immune system, the body's defense task force, is usually on the lookout for cancer. So how do these spies fall through the cracks? Well, we think it has to do with their camouflage. and that camouflage is made of sugars. Every last cell in our bodies is coated with a dense forest of sugars. We call them glycans, and they act a lot like the clothes we wear. Some are more flashy, while others give cells a more discreet look. And our immune system has a keen sense of fashion. It can notice these patterns and respond when the wardrobe change feels off. But cancers can be quite clever. They will tailor their outfits, tweaking their sugars to blend in. And my work is dedicated to understanding how this camouflage can help cancer cells evade our immune system. First, we want to understand, what does this camouflage actually look like? What are the molecular patterns that we find? Secondly, how is it helping cancer escape our body's defenses? And finally, if we strip them of their camouflage or train our immune system to better recognize it, can we enable our body's defenses to detect and eliminate cancer cells before they wreak havoc? By understanding how these cells hide, we might stop it from ever getting to that point. We might be able to prevent instead of having to treat again. And for people that live with or who have beat cancer, that doctor's appointment in the future might just be a routine checkup. No waiting and wandering, no second battles in the horizon, just peace of mind.
Okay. I would like to start by asking you this question. Do you know the type of cancer that kills the most? I'll tell you, it is lung cancer. According to the World Health Organization, lung cancer was responsible for over 1.8 million deaths globally in the year 2022. This is because the disease is largely asymptomatic until late stage, leading to late diagnosis and low survival rates. However, with screening, this disease can be detected early so that the patient can start treatment early enough and have a higher chance of surviving the disease. And that brings us to the core question of my PhD research. What is the feasibility of recruiting participants for lung cancer screening from occupational settings? And our methodology is based on 3 R's. One is review, recruit, and reflect. We started out by conducting a systematic review of literature on recruitment of employees for lung cancer screening across different occupational settings. And then we went on to conduct a pilot recruitment from 3 German companies into an ongoing lung cancer screening project. And then lastly, we invited these employees who participated in the screening and the occupational health physicians who facilitated their recruitment for interviews. Our review showed that research and implementation of lung cancer screening in occupational settings is still minimal. However, we also found out that the employees can be recruited by emails, through letters, through face-to-face interactions, as well as through their heads of departments. 79 employees participated in our pilot recruitment. And out of these 79, 26 consented to be contacted again for the interviews. And at the end, 17 of these employees participated in the interviews, as well as 3 occupational health physicians. Now we are currently undergoing analysis of these interviews. In Germany, at the moment, it is being planned to start a national lung cancer screening program. It is expected to start by April of next year. So it is our hope that our study, particularly this, my PhD research, would be instrumental in ensuring that employees, especially those who are exposed to the occupational risk factors of lung cancer, can participate in the national lung cancer screening program in Germany when it starts next year. Thank you.
Imagine in your everyday life experience smashing waves, powerful winds, and strong temperature shifts. That's what, for example, what a mussel experiences in its everyday life. Coastal regions are very tough ecosystems where species are in a constant battle to survive under daily conditions. But not all coastal regions are the same. My work takes me to northern Norway above the Arctic Circle, where it's also very complicated to live. My work is specialized in blue mussels, and I'm trying to understand how freshwater input can affect freshwater. If you don't know, northern Norway has the second largest coastline in the world, but at the same time, it's very understood because Arctic coastline is also one of the strongest affected by climate change, and we know very little about it. And this is exactly why I'm trying to understand how strong environmental stressors that is also very important affect blue mussels. Did you know, if you've ever been to Norway, you also probably noticed how mountains seems to touch the sea, and they are covered by snow melt and glaciers that all arrive to the sea. This freshwater input can extremely change the environment that we are surrounded by. But as we see in the picture, all the mussels are settled inside the freshwater, and we just said that it's a very stressor from them. Why is this? Because their main predator, sea snails, are unable to survive under these conditions, and they cannot crawl inside the freshwater to eat the mussels. So mussels can use it as a refugee from the predators. And why is the take-home message here? Environmental stressors can definitely shape an ecosystem. In our study, just taking one step outside the freshwater area takes us to a new, different world, where all of the species that we find are different from what we find inside the freshwater. So if this happens in a small area of 100 meters study area, how will it be on a larger scale when temperatures rises and freshwater melt will increase in the Arctic? How will species like the mussels survive, adapt, or fail to adapt under these rapidly changing conditions? Thank you.
How do you like your eggs? You see, it's an important question. How do you like your eggs? Some people like them boiled, scrambled, fried, sunny side up. If you're feeling fancy, you might like them poached. There are no wrong answers to the question, how do you like your eggs? Unless you say raw. Now, what if I tell you there is one more method of cooking these eggs? If you could mix them hard enough or fast enough, like in a blender. Now, you might ask this question like, why would that cook eggs, right? And when scientists looked deep into the mysteries of the universe, they found out that just like the Constitution of the US, all energies were created equal. And the energies of the universe, for the universe, by the universe, shall not be destroyed, but merely converted. This is what the science nerds call the first law of thermodynamics. But you and I, let's test this theory out. Take your right hand with me and start moving it in a circle. Come on. And take your left hand and bang. Again. Faster. Thank you. What you just did is you converted the energy of movement to the energy of sound. And what I just did is slightly manipulated you to clap for me. But in the same way, in a blender, this energy of movement can convert to energy of heat, which will cook these eggs. Which means that if you have really strong muscles, you can actually whisk your eggs hard enough to cook them. But then you need cooked eggs to make those muscles in the first place. And this is the circle of life. And in this circle of life, we are made up of really tiny cells, the primary unit of life. And these cells, just like us, they catch diseases, they age, they die, and they sometimes cannot be replaced by our bodies. Which makes us wonder what if scientists could make artificial cells that were better at doing some things and that could do what we want them to do when we want them to do it. Now, of course, when I say artificial life, it sounds shady. But our relationship with life has always been of a shady one. When life gave us lemons and grapes, we made tinto de verano, the ultimate form of the two. When life gave us millions of years of evolution into a horse, we were just not happy with the amount of horsepower it generated, and we created cars. In the same way, scientists have started developing synthetic cells that could actually replace cells in biology and could actually help us determine what we need to do at certain spots. Now, unfortunately, these synthetic cells are not really alive, and they also do not have energy. So we developed really tiny motors, 1/1,000,000th of a millimeter in size. These really tiny blenders can be run with light, and they can provide energy to these cells. And with using this energy, we showed that they can actually change morphology inside living systems, which is actually crucial in biology. So in the not so far future, you can use these synthetic cells to build your muscles to cook your eggs, and when someone asks you, how do you like your eggs? You can answer, blended.
It's not, okay. Hello everyone. So now from the eggs, let's move on to Africa. Let's all in your imagination, let's go to Africa, Namibia. So once I say the word Namibia, what comes to your mind? Lots of animals and too hot. It is even hotter than this Spanish region. So if you consider on the first image of this slide, which is problem and experiment, the problem is in Namibia, we have no water, no rain, too hot, extremely hot, 55 degrees of temperature. And the sand and the soil in which you grow plants is the worst kind of soil that ever exists, which is sandy soils. And I know with experience, some of you here have always killed some plants in your house. So if you don't water plants, how will plants grow? So this is my question that I will going to discuss it right now. So in the first image you see, because of all these environmental problems, there is a creation of dry soil layer. What happens is that water is stuck below ground, which is below 50 centimeter, and this cannot come up. But the temperature is so high that it boils at 50 centimeter below the ground. So only vapor can travel up. Now my study is, can plants survive on vapor? Most important 3 words: sand, no water, super hot. Can plants take up water vapor? We brought back everything from Namibia in Hamburg in my lab. And then we did an experiment here. Everything the same. The same plant, the same soil, too much high temperature, low humidity, no water, no irrigation. Only thing I changed is this red water that you see. I added a marker, a label. So when the molecules move from below to the up, I can see where you are going. It's like a GPS tracker. So on my husband or something like this. So I know where he's moving. So all the molecules, I know where they're moving. Now let's come, what was my results? You can see in the figure, in the third figure, in the results section, you see where the molecules are. So molecules did move from below groundwater as vapor to the soil, here, here, and in the root zone. So plants did take up water vapor. I repeat, water vapor. They did take up water vapor, and it went all the way to the leaves. So that means plants can take up vapor. It survived for 2.7 days, and then we did a little manipulation, let's say. I added a plastic layer, and with that, it reached to 4.5 days. Now, what is the impact of this? We have new knowledge. In the whole wide world, nobody knows if plants can survive on vapor. So we are one step ahead of them. I do not know how do roots take up vapor, but we know that they do take up vapor. And then we have now better knowledge. How does it impact African farmers? For you, it might not be much, but this creates a less panic, because when the water tanker is coming, they know that they can wait for 2 days. So there is less panic, and which is much more important in real world scenario, I would say. So sand, water vapor, and plant. Plant can do survive on vapor. Thank you.
Okay. Don't worry, it's not a serious cough. It's, sorry. Don't worry, it's a normal cough. But what if it became more serious problem, or it carries tuberculosis droplets? TB is a airborne disease, meaning a single cough and sneezing is enough to spread. And around 10 million people develop tuberculosis worldwide. So the bacteria cells come in the lungs and our immune cells try to trap and destroy it. But TB is clever. Instead of dying, it stays inside the lung cell for years and wait potentially and living potentially. And though there are some anti-TB drugs, but they have severe side effects like liver damage, nausea, even vision loss. On top of that, the treatment is longer, up to 2 years, and TB develops resistance to these drugs. So, what can be the solution? In my thesis, I designed a novel strategy loading the anti-TB drugs inside the nanoparticle, which are super tiny, only few nanometers. Do you remember the Greek mythology where the soldiers were hid inside the Trojan horses? So similarly, my nanoparticle act as the Trojan horses and deliver directly to the infected cell and crush TB from the inside. And do you want to know my another weapon? So I am using a special kind of stabilizer that has 3 powers. It stables the nanoparticles and it carries the drug inside its little pocket. And the best thing is, it kills the bacteria all by itself. So really, a true 3-in-1 superhero. I have no doubts you are curious about my results. Well, I successfully packed 100% of drug inside these nano Trojan horses. So my tiny soldiers are ready to move forward and leaving no escape for TB. So no more cough and no more side effects, only my smart nanoparticle. So together we can fight against TB. Thank you.
Thank you. Can you recall a time in your life when you suffered serious hunger due to the unavailability or scarcity of food, or perhaps lost your investment as a result of unfavorable weather conditions? Sweet potato is a globally significant food crop and also feed crop that currently ranks number 6 among the most important crops in the world. Conventionally, it is propagated using stem cuttings and part of the roots. However, this method is number 1, relatively slow. Number 2, it produces unhealthy propagules that cannot withstand unfavorable weather conditions. Therefore, micropropagation has been adapted as a salvable technique to remedy this situation. My research focuses on assessing the effect of plant growth regulators on sweet potato micropropagation and also their response to drought and cold stress. In the process, nodal segments from sweet potato vines are sterilized and then cultured on hormone-supplemented growth media at varying concentrations. The regenerated plants from the growth chamber are transferred into pots, and then the pots are acclimatized and then transferred to the field for tuberization for about 4 to 5 months. The CRD, which is the completely randomized design, and then randomized completely block design are the common experimental designs used in my work. Statistical analysis of data collected are being performed using R and GenStat. It has been found through my work that the application of 1 milligram per liter of benzoaminoporine, a synthetic cytokinin, can enhance the node number, leaf number, and chlorophyll content of sweet potato. Also, it has been found through my work that application of 5 milligram per liter of the same hormone can enhance the resistance of or tolerance of sweet potato to drought stress. Finally, it has been found through my work that different genotypes of sweet potato respond differently to different types of plant growth regulators. What is the findings of this work? The findings of my work hold significant relevance to SDG 2, which is Sustainable Development Goal 2, which is the achievement of food and nutrition security. Thank you very much.
The psycholinguist Frank Smith said, "One language sets you in a corridor for life. 2 languages open every door along the way." But does speaking 2 languages really make a difference? Well, as it happens, it does. But before explaining, let me take you to an ongoing issue in education, the achievement gap. This gap is the difference in school performance between children from high and low socioeconomic backgrounds, also known as SES. It has been, well, before explaining, imagine you were 6 years old. Imagine walking into your very first classroom, crayon full of dreams and backpack full of crayons and dreams, but already at a disadvantage, not because of your talent, not because of your effort, but because of circumstances beyond your control. Some doors are open to you and others are, well, closed. Studies show that children from families from low SES backgrounds begin primary school and move into secondary school with less developed executive functions. These are the cognitive skills that help us remember information, focus, and switch between tasks, essential for academic success. And this is where bilingualism comes in. Studies have shown that learning more than 1 language helps you develop into and strengthen precisely these executive functions. So the main research question of my thesis is, can bilingualism close this gap by developing the executive functions of children from low SES backgrounds? So to find out, I worked with 49 students from year 3, 7, and 10 from the bilingual program CLIL in 2 primary and 2 secondary schools in Madrid. Each student completed 3 cognitive tests to measure the 3 core executive functions: inhibition, working memory, and cognitive flexibility. Now, the results were striking. Among children from low SES families, working memory, the ability to hold and use information, increased significantly from year 3 to year 10. In fact, their growth was stronger than that of the high SES peers. Why is this important? Because working memory is one of the strongest predictors of academic success. And what does this mean? This means that learning more than 1 language, that a bilingual education builds cognitive tools that give disadvantaged students a real chance to thrive. So I invite you to imagine the possibilities. If every child, disregarding the background, has access to a bilingual education, we wouldn't just be teaching them words in 2 languages, we would be giving them tools to open doors to opportunities that might otherwise remain closed. Because imagine that 6-year-old walking into the very first classroom, no longer at a disadvantage, but stepping into a world full of possibilities where the potential truly can flourish and where every door in that corridor is open. Because in the end, bilingualism isn't just about speaking 2 languages. It's about giving every child the opportunity to succeed. Thank you very much.
Good afternoon. I am Zara from the Pharmaceutical Analysis Department. In the Pharmaceutical Analysis Department, in my thesis, we are developing analytical method for analyzing and quantitation of compounds that is exogenous, which means it's outside from the body, produced outside like synthetic, or endogenous, it's inside of the human body, and it's found in biological samples. Biological samples can be of plant origin or from animals or from humans like plasma, urine, etc. But we cannot actually inject it directly in the instrument. We need to do some sort of purification to get our compound in the sample preparation procedure. And after that, we will take it to the instrument, which is ultra-high performance liquid chromatography tandem mass spectrometry. So in the liquid chromatography, we are basically doing separation for our compound from the mixture. And we are getting the component of this, the compound or the component that we are looking for, as you can see in the chromatogram, depending on the retention time. In the mass spectrometry, we are doing fragmentation implementation of these compounds so we can get the quantifier ion that can help us to quantify precisely our compound and the qualifier ions. And then we do data analysis and statistic. Up so far, we answered so many important questions, medical questions. So the first question that we answered is, should we prescribe nimodipine before or after a stroke? Actually, it can be prescribed before or after or even during the stroke because the pH of the brain area is not actually changing. It's not affecting the nimodipine concentration. And regarding quinuric acid or quina, it's endogenous compounds found in the human body, and it's involved in so many diseases of the CNS or central nervous system, but cannot be prescribed outside of the body because it's not permissible to the brain. And we investigated the permeability of quinuric acid derivatives with the same exact biological effect. And we also quantified very important compounds from plant origin like stilbenes and phenylethrines. In the untargeted analysis, this is in the targeted analysis. In the untargeted analysis, we are investigating the component of the human plasma, and we are also investigating new compounds produced by the bacteria under stress conditions. In the photo, you can see the spectrum and the chromatogram. That's how we present our results. Actually, it takes like months of work. And the bottom line that we cannot actually study any drug, and we cannot understand how the drug actually interacts inside of the body without a precise and sensitive and very accurate analytical method or LCMS method. So it's involved in the pharmacokinetic studies, pharmacological studies, toxicological studies, and all sorts of the study. And also it's in medicine, environmental science. So it's not only like pharmaceutical. We can analyze anything. Thank you so much.
Have you ever thought about what baby salmon go through in fish farms? Just like human embryos, they are vulnerable, especially to water quality. Imagine a salmon egg rest at the bottom of a gravel bed, tucked in a flow of a cold river. It's just days before hatching, unaware that its environment is about to shape its entire life. Now imagine that same egg in a hatchery tank with clean water, monitored, but not perfect. There can be problems. An invisible threat can creep in slowly. Ammonia. It's a natural byproduct of protein metabolism, but in the wrong dose at the wrong time, it can spell trouble. That's where my research begins. I want to explore what happens when salmon is exposed to ammonia in their earliest moments and how this shapes their ability to survive, grow, and thrive as adults. At the heart of my research are ammonia transporters and rhesus proteins. They are molecular gatekeepers that regulate internal ammonia levels. But in developing embryos, these systems are still waking up. So what happens if we force them early into action? When salmon is laying the foundation of their future resilience. That's what I'm testing in the lab. I expose salmon in different developmental stages to sublethal levels of ammonia and monitor what happens on the inside. I look at how these transporter genes turn on, how stress hormones spike, and more importantly, how epigenetic markers are laid down. You know, epigenetics is like nature's memory system. Chemical marks on DNA that tells genes when to speak up or stay quiet. If we stress or stimulate salmon early, we may be writing a molecular story that they may carry for the rest of their lives. A developmental programming. And here's the twist. Sometimes this story can make them stronger. What doesn't kill you makes you stronger, right? A little stress at the right time may program salmon to become more robust adults. It's called hormesis, low-dose stress leading to greater resilience. But if the timing or dose is wrong, we may lock in vulnerability instead. Slower growth, weaker immune system, or even poor multiplication and survival. That's why this research matters, because by understanding early life programming, by literally writing better beginnings for salmon, we can improve their health, reduce loss in hatcheries, or even raise fish that are better equipped to handle the stressors of a changing climate and modern aquaculture. In short, we're not just raising fish. We are programming resilience one salmon at a time. What about you? Were you programmed to better handle stress? Thank you so much.
Let's imagine for a moment that your body is like a living city, where every part is constantly sending signs about what is going on inside. Now, what if I told you that a small sample of urine could reveal a hidden danger, like bladder cancer, even before any symptoms appear? In my research, I take on the role of a detective. But instead of searching for suspects, I'm looking for clues. Tiny chemical clues in urine that might indicate early signs of cancer. These clues come in the form of volatile compounds, molecules that easily evaporate and carry important messages about what's happening inside our bodies. Think of these compounds as invisible hints left behind at the scene. On their own, they might seem small or insignificant, but when put together, they form distinct patterns. And just like every single one of you has a unique fingerprint, our body carries its own chemical signature. That's why I like to say, urine is our body's fingerprint of health. To help explain, let me share a fictional example. Imagine John, a 55-year-old man, who understands the importance of regular health checkups. During one of his visits, his doctor asks for a simple urine sample. In the lab, instead of searching for blood or cells, we analyze the invisible fingerprint left behind by volatile compounds. If the pattern I've been studying is found, one that appears more often in people with bladder cancer, this might act as an early warning sign. This is the goal of my research, to develop a method to detect bladder cancer early, using a simple, small, and painless sample, by decoding the body's own chemical clues. This could one day help doctors act sooner, avoid unnecessary and costly procedures, and give patients a better chance of survival. Bladder cancer is often called the forgotten cancer, despite being one of the most expensive cancers to treat and prone to coming back. But I believe that by becoming better detectives and learn how to read these early signs, we can stop treating it too late and start catching it just in time. Thank you for following me in this journey. And remember, every drop of urine holds information, and with the right tool, it might hold the key to saving lives.
So let me start with a quick question. Who here has heard of Oktoberfest? And what's the most important thing for Oktoberfest? Beer, right? Can you imagine Oktoberfest without beer? That would be devastating, right? Now, believe it or not, this nightmare could actually come true, because beer is made out of barley, and barley has a tiny invisible enemy called Plumeria hode. Sounds fancy, scientists use Latin names to sound smarter. Now, Plumeria is a fungus, and this causes powdery mildew, this white powdery stuff that you see on the leaves. It looks harmless, right? But it's not. It's a plant killer. It's responsible for huge crop losses every year. And what do you think makes Plumeria so dangerous? It's not just sitting there chilling on the leaf. It produces an infection structure called Haustorium. It's like a feeding straw that enters the plant leaves and steals the plant's nutrients. It's basically biological looting. And barley tries to fight against this, but it needs help. This is where I come in. Think of me as a spy, and my weapon of choice, a really powerful microscope. I try to study these fungal tunnels and map how Plumeria breaks in. Now, how do you spy on something that's so tiny? Electron microscopy. I take infected barley leaves, process them into resin blocks, similar to the mosquito from Jurassic Park. And I take these resin blocks, make them thin into thin sections, like 70 nanometers, thinner than a strand of hair, and then I zoom in more than 10,000 times. I don't just make 2D pictures. I make a 3D model of this infection structure. And why 3D? Because with 2D, it's like trying to understand a battleground with just one picture. But with 3D, you're flying into the war zone. You see where the fungal tunnel's in, how the plant walls bend or break, and how the defenses are overrun, because this is how we find the weaknesses and plan a counterattack. because in every battle, strategy is key, and barley needs allies. It's not just about seeing, it's about understanding. It's about understanding where the fungus strikes, how the plant responds, and where we need help. Because here's the deal, you cannot stop something that you do not understand. By understanding barley's attack, we're not just saving barley crop losses, we are protecting food security, and of course, making sure that there's beer at Oktoberfest. So next time you raise a glass of beer, remember that there's a tiny war behind every sip. And I'm working really, really hard to make sure that barley wins this war. Thank you.
Hello. Today I would like to share with you one of my research projects which is focusing on the protecting the blood-brain barrier after the ischemic stroke attack. Our brain has a very specific and important protective structure which is called blood-brain barrier. It's formed by very, very tightly connected cells which is located in the wall of the brain blood vessels. It works like a security system which can blocks almost all the harmful substance from getting to our brain, but during the ischemic stroke attack, the flow to our brain will be blocked by the thrombus. In that case, both of the brain tissue and the blood-brain barrier will be break down. Because of leakage of the blood, the breakdown of the blood-brain barrier will make the many harmful substances leak into our brain tissue. That leakage will worsen the injury and makes the recovery very slow. So here's where comes our question. Can we protect our blood-brain barrier after a stroke attack? To study this one, we established a cell culture mode of the blood-brain barrier. We seeded the human brain endothelial cells together with the supporting palisates, which are the most important two cell types of the blood-brain barrier. We simulate the stroke by cutting off the oxygen and glucose supplies for 6 hours. And after that, we let them to rest for 24 hours. During the recovery part, we tested one medication, which names as SHAHA, or another name, volinostate. We would like to see if the volinostate can bring the positive effect on the blood-brain barrier. We measuring the two most important parameters of the barrier functions. First, the electricity resistant across the barrier. And second, the permeability of the molecular markers. And as a result, we found out that the SHAHA indeed increased a lot of the barrier functions by increased electrical resistance, which means the barriers are more tighter, and to decrease a lot of the permeability of the molecular markers, which means that the barrier are less leakage. As a result, we can see that the SHAHA indeed protect our blood-brain barrier in the cell culture mode. Although we are just in the really early stage of the laboratory experiment, but our results suggested that the SHAHA might help the patient to recover from the stroke attack by reducing their brain barrier functions damage. Thank you so much for your attention that I would like to share for you.
Every one of us here has driven a car, eaten at a restaurant, listened to a class, or played a game on their phone. What if I told you that all of these things, even though they're completely different, that the processes that make them are essentially the same? In the 1950s, after World War II, Toyota needed to survive the market, and they started to develop something called the Toyota Production System, a system aimed at improving processes through elimination of waste in them and continuously improving them. This system enabled Toyota to become the global leader in the world and hold the position even today. Of course, competitors noticed the success and they wanted to copy it. Not only in their industry, but also companies in other industries. So what they did was they tried to implement it in their way of working, but majority of them failed. Why? Because they pick and chose which part of the system they would implement, and there was no clear structured methodology on how to implement the way that Toyota works. What resulted is in a series of methodologies that only were able to improve processes, but not sustain and continuously improve those processes. And this is where my PhD focuses on, developing a structured methodology based on Toyota's way of working that not only ensures that you set up the process correctly, improve it, but ensure that you improve it continuously over time. And I call this methodology the infinite development loop. My thesis aims to answer three key questions. The first one is it possible to develop such a methodology? The second one is there a positive impact from implementing this methodology to process performance? And the third one is it applicable to other industries? My focus industry is the automotive, and I can say that the answer to all three questions is yes. I've applied this methodology to certain processes in the automotive industry, and the results have shown an improvement of process performance by up to 75%. Thank you.
Imagine this. You walk into a classroom. Students are swiping, clicking, searching faster than you can follow. And in the front stands teacher, still unsure how to use a half of the tools available. This is not a distant scenario. It's reality in many schools in Serbia. And the gap is even more striking in geography, a subject that could come alive with interactive map, real-time data, and even virtual field trips. So I ask myself, how ready are geography teachers in Serbia for the digital age? And what about future geography teacher, final year students in our universities? To answer this question, I surveyed more than 200 geography teachers and over 100 final year students using European DigComp Agile framework. And here are of the some results among the teachers. 50% are beginners, 26 are explorers, 29 integrators, 70 experts, 11 leaders, and only 2% are on C2 level like pioneers. And the biggest weaknesses of the research were not technical, but teaching skills of the teachers. Teachers struggled most with tracking progress of the students, supporting independent learning, applying digital tools in teaching. And what about students? Students were confident with tools, with creating contents and using devices, but in real teaching, the same gap appeared. So message is clear. If we want true digital transformation in education, we must start first with our teachers. We need subject-specific training with practical skills, including interactive maps, geospatial tools, and collaborative platforms. So if we want our students to understand and change the world, we must first empower teachers who guide them. Thank you.
Thank you. My research is on teacher resilience. So I will start my presentation by asking you a simple question. So have you seen a tree standing strong in a storm? So look at that tree with so many deep roots, which sustain that tree to be strong against heavy rains, stronger winds. It can't fall because it's very strong. So imagine this tree as representing that teacher who faces daily challenges like overcrowded classrooms, limited resources, heavy workloads, emotional exhaustion, and high demands, and so many challenges. So, but I can ask ourselves, why do some teachers remain resilient and keep enjoying teaching while others struggle? That's where my research comes on. So my research focuses on understanding what keeps these teachers thrive while others struggle. So in resource-constrained environments like Rwanda, that's where my research is based on. So teacher resilience is key to teacher well-being, retention, and overall educational outcomes. So by mixed methods designed through surveys and interviews with hundreds of teachers, I'm identifying both personal factors and external support systems that sustain those teachers. So we found that resilience is multidimensional. Just like those deep roots, resilience is built through a combination of personal factors such as confidence, coping strategies, and external support systems from school leaders, colleagues, families and friends, students, all of which sustain them to stay strong in their teaching profession, to stay motivated, keep teaching, and stay in the teaching. So by concluding, when we can ask, how do these teachers keep going? Just like those deep roots, teacher resilient, who are resilient are found to have those deep roots which sustain them. So by identifying and strengthening those roots, our research will help schools and policymakers to create that conducive environment where teachers are well supported and comfortable to continue shaping the next generation without burning out or without leaving the profession. So thank you very much for your kind attention.
Well, I was saying that we had another student, but she was not able to make it here. So this is the end of the event. Now the panel, the board, has to think about their verdicts. So thank you very much, all of you, for your presentations, for your pitches. They were all fantastic. I can give my opinion because I am not a member of the jury. And now we are all going to rest until the jury has their verdicts. Thank you all.
So hello everybody. Finally we agree before we say the names that we give some general feedback, of course, because I mean, as you already know, I mean, they were fantastic and very interesting, and yeah, for us it was a hard decision, and every presentation was very individual, original, of course, because you are individual and special, and I think this is one of the learnings, perhaps your own experience, but also how you saw your like your peers, that it makes such a difference, you know, if you just see the slide or if you hear the people talking. So I guess this is something we can all take home from this. You matter, you know, in science communication, you as a scientist, you as a communicator matter. So we would like to encourage you, you know, to, however, if you won or if you haven't won in this competition, you know, don't leave science communication. And yeah, try to get more experience in different settings. Of course, I mean, this is a special setting we have here, but you know, there are many options where you can do science communication in your university, but also in museums or in your city or elsewhere. There are lots of public engagement formats where you can, you know, get into contact with different audiences. So what can I say? Of course, I would like to, you know, engage with every one of you and give a single feedback, but we don't have the time for that. Also, the time was very short for that. But we realized as a jury, we talked a bit about that also in our short session before. Many of you use metaphors, you know, pictures even, and yeah, but also like metaphors we have, like war or criminal investigation or something like that. And we do like that, of course, because these are concepts known to the audience, and it helps incredibly, you know, to build a bridge between your specific area of research and the world of your audience, which has in most of the cases nothing to do with what you are doing every day. So do use metaphors. It's really good. It helps. Also, be careful with the metaphors you choose, because some of the metaphors, have two sides. Of course, they are interesting and, they draw the attention, but also sometimes they can create fear or, bring along some emotions that might be a bit critical for your audience. So just be careful with that and try it perhaps also. What do you associate with these things? They're interesting, yeah, but also perhaps they can create an image of science that might be a bit, what are they doing there in the lab? Are they creating some kind of monsters, or is this science out of control or so? But yes, also with the metaphors comes the use of images. You were encouraged to use images in the slides, and I think this is really great. We saw great examples also. Just as you saw, sometimes it's just one picture or two or three, and this really helps to anchor the information you got and combine it and link it to an image. Some of you experimented with ChatGPT/AI generated pictures. Sometimes it worked really good. Sometimes perhaps you also noticed that was perhaps a bit too not enough, not clear enough. So let's see what the technical progress brings with that. I think it's good to make more experience with it. Certainly it can help, much better than some of the technical images you used. Some of you had more like poster presentations with lots of information and technical terms. Be careful with that, because in 3 minutes you cannot relate all your talking to the images you show. So really try to use just a limited number of images that your audience can really process in this short time they have. And lastly, perhaps also, and I know this is very difficult when you have to concentrate and focus on your talking, try to keep the eye contact with your audience. It's incredibly hard, Some of you unconsciously move towards the slide, And this is where you lose the audience. So really try to focus on the audience. Also find a spot where you are, where you feel comfortable. Some of you moved. This is not a bad idea. You can move, of course, but not like a tiger, in a cage. Of course, sometimes it helps calming down and regulating your own stress level, but it can appear, it can make your audience feel a bit nervous. So try to move, but in a slow and relaxed way. And I think this is, I hope this general feedback helps you a bit. And I don't know if my colleagues want to add something.
Yeah. I can. Okay, I can add something. I subscribe to everything that was being said by my colleague of the jury, which is completely. Some of those things are actually important for you as you develop as communicators. Of course, this is a very special type of competition, and you have very straight rules, and you have always to adapt your presentation to the type of presentation that you're doing. So this is one of the things that all these things that Michael was telling, they are true, but you have to adapt them. And this contest brings up one subject which for me is very dear, that you have to communicate what you're doing, not only for your peers, because you have people from very different subjects, but to people from other nature and from other origins. And I always said to my students in one of the courses I give that if you are not able to explain what you're doing to your grandma, then you're not doing right, okay? So I think the big message is that you have to explain it to the audience that you have in front of you. You have to learn how to recognize the audience and then how to speak with the audience. Everything she said is true. You have to keep eye contact. You can move, but you shouldn't do some things. But always be aware of the audience that you are speaking. So one of the most important things, even when you go to a congress, when you do an outreach session with students from high school, always get to know who is going to be on the other side, and always prepare your speech to these people on the other side. Of course, here this was a competition that was a little bit different, so you are more tense, and you are almost competing among each other, so we have other things to think about it, but the most important is to know the audience that you're speaking. This is, I think, is probably the message most important to them. And all the rest was said here is good. You were all, I mean, all great. It's some of you were nervous. I don't know if one was coughing on purpose and even the cough was not coming right, but this makes part of the game, okay? So it's no problem. So we were able to understand all these small differences between you, but it was at least for me, was very rewarding to be here and to see how you, I learned some things actually. I took some ideas to my courses, so that would be also good. So thank you very much. You're giving me good material to work on it, so that was good. And congratulations. Okay, I think there will be a winner, a runner-up, but you were all fantastic, and it was really an honor and privilege to be here and to listen to you all. So that's my message. I don't know if you want to add something.
Someone has to do it.
Nothing else. Yes, I'll pass.
Thank you. And now we are going to tell the name of the runner-up and the winner for this EUGLOH 3 minutes PhD pitch competition. But first of all, congratulations to all of you for your research and your speech, because all of you are winners for being here. And the name of the runner-up is Andrea Badu. And the winner, Angela da Costa. Congratulations again.
Well, thank you very much. This is the end of today's session. Any practical information that you want to share?
Just one more minute. It's for tomorrow morning. If you remember, we need to go to the external campus. So the meeting point will be in the rectorate building, in the main door. So you will have to be there at 8 a.m. And then you will go with Ruth to the train station to go to the campus. Okay? So remember, at 8. And that's all. Thank you so much.